Alright, and now let's take a look at the paper discussions, the last part of the paper, and then I'm going to start summarizing everything. Now let's go over the paper's discussion section to see how they summarize their findings, or the quote findings. So they state that we report a novel COV, the 2019 nCoV, that was identified in hospitalized patients in Wuhan, China, in December 2019 and January 2020. Evidence for the presence of this virus includes identification in bronchoalveolar lavage fluid in 3 patients by whole genome sequencing, direct PCR, and culture. The illness likely to have been caused by this COV was named novel coronavirus infected pneumonia, NCIP, and complete genomes were submitted to GISAID, and phylogenetic analysis revealed that 2019 nCoV falls into the genus beta coronavirus, which includes coronaviruses SARS-CoV, bat SARS-like CoV, and others discovered in humans, bats, and other wild animals. And we report, quote, isolation of the virus and the initial description of its specific cytopathic effects and morphology. And direct PCR right here, because it was detected by direct PCR, refers to using PCR directly on the sample without prior DNA extraction or purification steps. Alright, and now continuing further into the paper, so going here, some molecular techniques have been used successfully to identify infectious agents for many years, and unbiased high-throughput sequencing is a powerful tool for the discovery of pathogens. Next-generation sequencing and bioinformatics are changing the way we can respond to infectious disease outbreaks, improving our understanding of disease occurrence and transmission, and accelerating the identification of pathogens and promoting data sharing. We describe in this report the use of molecular techniques and unbiased DNA sequencing to discover a novel beta coronavirus that is likely to have been the cause of severe pneumonia in 3 patients in Wuhan, China. So basically, they look at some patients, test for known sequences, and then they show that okay, it's not there, and then they're gonna test for the sequences that are present, and then be like, all right, we're gonna somehow use a computer algorithm and form a full sequence, say this is a new virus, and assume that's the cause of the pneumonia. All right. Going further here, this bioinformatics, I'll fix this up. Yeah, here, just fix that up. So bioinformatics is the field that develops methods and software tools for understanding big sets of biological data. Yeah, that's basically big data for biological systems. And continuing further in the paper, so although establishing human airway epithelial cell cultures is labor intensive, they appear to be, they appear to be a valuable research tool for analysis of human respiratory pathogens. Our study showed that initial propagation of human respiratory secretions onto human airway epithelial cell cultures, followed by transmission electron microscopy and whole genome sequencing of culture supernatant, was successfully used for visualization and detection of new human coronavirus that can possibly elude identification by traditional methods. All right, visualization just shows some blurry particles. Further development of accurate and rapid methods to identify known respiratory pathogens is still needed, and on the basis of analysis of 3 complete genomes obtained in the study, we designed several specific and sensitive assays targeting the ORF1ab, N, and E regions of the 2019 nCoV genome to detect viral RNA in clinical specimens. And then they would send these sequences and assays to the world, and they would basically go and start testing for these this new virus. Quote virus. So the primary sets and standard operating procedures have been shared with the World Health Organization and are intended for surveillance and detection of 2019 nCoV infection globally and in China. And more recent data show 2019 nCoV detection in 830 persons in China. Interesting. So they've already started testing people. Although our study does not follow Koch's postulates, our analysis provides evidence implicating 2019 nCoV in the Wuhan outbreak. And note here, so basically it just says it doesn't follow Koch's postulates. In Koch's postulates are 4 criteria designed to establish a causal link between a microbe and a disease, and are listed below. So the number 1 is a microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. And the microorganism must be isolated from a diseased organism and grown in pure culture containing only a population of that microbe. But for supposed viruses, they need a host. So that's a modification of the Koch's postulates. So the cultured microorganism should cause disease when introduced into a healthy organism. And the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. And basically to summarize, you gotta have to find a microbe in abundance in all the organisms suffering from the disease, but not in the healthy organisms. But remember, with viruses and even some bacteria, you can have asymptomatic carriers, which you can have that microorganism in healthy organisms, which eludes reasoning. And also then once you isolate it from the disease, it has to be grown in pure culture. And then the cultured organism should cause disease when introduced in a healthy organism. And then you should be able to re-isolate it from that and show that it's the same thing as the original one. That's Koch's postulates in a nutshell. And note that the postulates are not strictly followed anymore, and terms such as, quote, asymptomatic carriers, which are carrying the microbe but not the disease, and hence showing that there's a questionable causal link. And, quote, viruses which will require host cells to, quote, grow are assumed in mainstream science. So you assume a virus existence and you assume asymptomatic carriers. Yeah, so the asymptomatic carrier, this is cancels out step 1. And then step 2 is the viruses. So you can have Koch's postulates as originally formatted with viruses. Continuing further in the paper, additional evidence, presumably yet to be done, to confirm the etiological or cause of disease significance of 2019 nCoV in the Wuhan outbreak include identification of a 2019 nCoV antigen in the lung tissue of patients by immunohistochemical analysis, and detection of IgM and IgG antiviral antibodies in the serum samples from a patient at 2 points to demonstrate seroconversion, and animal or particular monkey experiments to provide evidence of pathogenicity. Of critical importance are epidemiological investigations to characterize transmission modes, reproduction interval, and clinical spectrum resulting from infection to inform and refine strategies that can prevent, control, and stop the spread of 2019 nCoV. Very interesting wording, because that's pretty much what's blustered for past few years during the COVID saga. Going further, so this 2019 nCoV antigen, right here, identification of that refers to the proteins or regions of proteins such as the, quote, spike protein, and on the, quote, virus that can serve, or the spike protein, such as the spike protein on the, quote, virus that can serve as antigens and, quote, stimulate the immune system. So those are the antigens, basically specific proteins or regions of proteins on the virus. I'll put this in bracket. Yeah, I'll put this in bracket like that, such as the spike protein. So the spike protein can be viewed as a antigen. And again, the spike protein, whatever, that's supposed to be the part of the solar corona around the virus, but that's questionable in my opinion as well. Going further, so immunohistochemical analysis, or IHC analysis, is a technique used to visualize and detect specific proteins or antigens within tissue sample. Yeah, so this is basically detecting the proteins and antigens and so on. Immunoglobulin M, or IgM, is the largest antibody and first antibody to appear in the response to initial exposure to an antigen. And immunoglobulin G, Ig, capital G, is the most common type of antibody found in blood circulation, and it represents 75% of serum antibodies in humans. And seroconversion is the development of specific antibodies in the blood serum as a result of, quote, infection or quote, immunization, including vaccination. So basically, you have antigen. And then seroconversion is when you build up these antibodies in the blood serum as a result of that antigen, and supposedly that the antibodies inside, they can go and neutralize that specific antigen. Again, that's the mainstream theory. So immunization is the assumed process of strengthening an individual's, quote, immune system after initial exposure to an, quote, infectious agent. And pathogenicity is a measure of how effective a pathogen is at causing disease. So yeah, basically they're saying they need monkey animal experiments and basically try to infect them with these supposed viruses, as well as other procedures that likely involve torture. Yeah, because when you read up on how they actually test on animals, it is pretty brutal. Going further, so transmission mode right here too, because they want to determine transmission mode of this supposed virus, is the specific way a disease or pathogen is transmitted to another person or organism. And then reproduction level is the average time it takes for an infected individual to, quote, transmit the disease to others. And this clinical spectrum is just a full range of signs, symptoms, and outcomes that can occur as a result of a, quote, disease or condition. All right, let's continue further. So summary of paper discussion of the paper's discussion. So let's go over a summary of the paper's discussion section and let's break it down. So a novel or new CoV coronavirus was identified in hospital patients in December 2019 and January 2020. Evidence for this, quote, new virus includes identification in lung fluid samples or more accurately, airway fluid samples by the following methods: whole genome sequencing, PCR directly on samples, and cell cultures from the samples. Complete genomes were submitted to the GISAID, and the, quote, new virus falls into the beta coronavirus classification. Modern genetic sequencing and big data analysis technology is quote, accelerating the identification of pathogens such as this, quote, new virus that is, quote, likely to have been the cause of severe pneumonia in 3 patients in Wuhan, China. Human lung fluid samples, with the assumption that they, quote, contain a new virus, were used to, quote, infect human airway epithelial cell cultures. Quote, virus particles were assumed to be visualized via an electron microscope. And whole genome sequencing was performed on the cell culture supernatant. From the 3, quote, complete genomes, the authors developed techniques to identify this, quote, new virus by targeting specific sequences from these genomes in the targeting of, quote, viral RNA in future patients. The sequencing and procedures were shared with the World Health Organization, the WHO, for the surveillance and detection of this, quote, new virus globally. As of the paper's publishing, 830 people in China tested positive for these targeted sequences. The authors admit their paper does not follow Koch's postulates of a more concrete link of microbe to disease causation. The authors instead claim their study, quote, implicates the, quote, new virus in the Wuhan, quote, outbreak. All right, so if you scroll back up to see that part, there's the Koch's postulates. So our study does not follow Koch's postulates. Our analysis instead provides evidence implicating 2019 and CoV. All right, so we have that implication. Going further, additional experiments need to be done to confirm the disease-causing claim of this new virus, and these are identify proteins on the quote virus that serve as antigens in the lung tissue of patients, detect corresponding antibodies or proteins in the blood of patients that form to, quote, identify and neutralize the, quote, virus antigens. So go look in the blood and look at antigens in the lung tissue. And animal, or particularly monkey, experiments to demonstrate the virus actually actually causes disease. And the paper concludes by highlighting the importance of epidemiological investigations to, quote, investigation to characterize the transmission and clinical symptoms of this quote disease in order to refine strategies to, quote, stop the spread of this new virus. Interesting. So yeah, I'm just paraphrasing that.